very small in size. That's what I told you earlier. The dystrophin, mutated dystrophin protein is short and it will not be able to bind to the dystroglycan complex. Consequently, the short dystrophin protein will not be able to fulfill its job. Bismillahirrahmanirrahim. My dear students, Assalamu alaikum wa rahmatullahi wa barakatuhu. Welcome back to AmreenaMehrajTutorials. My dear students, you already know that we are dealing with Mendelian single gene disorders, and this comes under the syllabus of class 12th. Questions could be asked from this particular topic. It's a very vast topic. Questions could be asked from this topic in your board examinations, coming board examinations, and the questions could be asked from this particular topic in your competitive medical entrance examinations, even in your paramedical entrance examinations as well. So my dear students, as far as Mendelian single gene disorders are concerned, I have already discussed two disorders under the category of X-linked recessive disorders, because Mendelian single gene disorders are themselves classified into four different categories. The first category is the X-linked recessive disorders. Out of X-linked recessive disorders, I have made you understand about hemophilia, its inheritance pattern, what happens in that particular disease, and I've taught you about color vision deficiency, what we usually call color blindness. Now today we are going to take up another important X-linked recessive disorder under the category of Mendelian single gene disorders. That's going to be the muscular dystrophy. Muscular dystrophy is a very vast topic, and some of the muscular dystrophies are not even X-linked, they are autosomal dominant. So if you are confused anywhere, you have found out muscular dystrophy in autosomal dominant category and you're confused that why do you find some muscular dystrophies in X-linked recessive disorders, today is an opportunity for you people to clarify your doubts and all. So as far as the board is concerned, you must be able that there's a muscle cell membrane over here. So the defect is basically muscular in nature. There is problem in the muscles. Okay, the problem in the skeletal muscle cells, the problem in the cardiac muscle cells. So the reason lies in the mutations at the genetic level, and that too the single gene mutation that results in these muscular degeneration problems, what we call the muscular dystrophy. So we are going to talk about this in detail, and inshallah by the end of the lecture you will be able to distinguish between different types of the muscular dystrophies, and you will have a strong hold on one of the most important type, that is called Duchenne's muscular dystrophy. So stay tuned, be with me, and take a copy and pen and start penning down every important point which I'm going to discuss with all of you people. Before starting explaining this, what is there on the board, I would request all of you to just like this video, comment on this video, share this video with your friends and colleagues, so that I'll reach to more and more students, and more students will join this channel of AmreenaMehrajTutorials, so that others will also be benefited. And don't forget to subscribe to my channel and press that bell icon so that you will be notified, because I'm planning to come with a live session. If you want to get notified for my live session, you have to press that bell icon so that you will receive the notification when I'll come live for solving your live questions as well. So let's begin the topic itself. Now my dear students, I have written over here muscular dystrophy, and no need to worry about the content which is on the board. It's very simple. Before starting explaining, I would like to make you understand about muscular dystrophy. It's a group of familial disorders. So muscular dystrophy is not just one disorder, it's a group of familial disorders. It's a group of familial disorders in which there is basically a progressive degeneration of the skeletal muscle fibers. It's a group of familial disorders that cause progressive. That means it's not an abrupt disorder. It will appear in an individual in a span of couple of years, maybe 5 or 6 or 7 years. The effects will be prominent. Progressive degeneration of muscles of the body, specifically skeletal muscle fibers. So not only skeletal muscle fibers are going to be affected or degenerated, even the cardiac muscles, the muscles of the diaphragm, which is the separation, which is the muscular partition between the thoracic cavity and the abdominal cavity, that's also going to be affected. Rather, saying affected, it's better to say degeneration, deterioration. In muscular dystrophy, basically the muscles start degenerating with the passage of time. The reason is genetic in nature, okay? Now, my dear students, these disorders, these muscular degeneration disorders are of broadly, I can give you the example of 3 major disorders. That's very, very important for you people. So, we have 3 types of muscular dystrophies. Number 1 is very important. Number 1 is Duchenne's muscular dystrophy. Duchenne's muscular dystrophy is very important for you people in class 12. Duchenne's muscular dystrophy. I'll write down MD. MD stands for muscular dystrophy, okay? Number 2 is Becker's. That's also very important for you people. Becker's muscular dystrophy. These 2 are important for you people. Third one is called the myotonic dystrophy. Very important as well. Myotonic dystrophy, okay? Now, my dear students, what do you people need to remember is that Duchenne's muscular dystrophy is X-linked recessive disorder. Okay, this is X-linked recessive disorder. Becker's muscular dystrophy is also X-linked recessive disorder. Very important for you people to remember. But this myotonic dystrophy is not X-linked. That's very, very important. This is an autosomal, not even recessive. This is autosomal dominant disorder. This is the first thing you need to remember. So these 2 have the same type of cause. The cause for these 2 is the mutation of X chromosome. There's particular gene which is located on the X chromosome that gets mutated, which results in either Duchenne's muscular dystrophy, what we call the Duchenne's muscular dystrophy basically, and we have the Becker's muscular dystrophy because of the X-linked recessive mutation. But myotonic dystrophy is not caused due to X-linked recessive mutation. It's an autosomal dominant disorder. That means in this case the mutation happens to the genes which are located on the autosomes. So let us over here try and remember which is the chromosome which is involved. Either you have the mutation on chromosome number 19 or you have the mutation on chromosome number 3. This is X, this is S, but this is chromosome number 19, chromosome number 3. That means you got an idea. Both of these are what? Both of these are which chromosomes? Are they sex chromosomes? No, they are autosomes. Okay. If chromosome number 19 is mutated, it's basically a gene which is present on this chromosome. I will give you the name of the gene. The name of the gene is basically DMPK gene. That is mutated. Okay, if this gene is mutated, the resultant disorder is called the myotonic dystrophy type 1. Okay, so you have myotonic dystrophy. This type of dystrophy is of 2 types. Myotonic dystrophy type 1, when there is a mutation on the gene, DMPK gene, which is located on chromosome number 19. If there is a mutation on a gene which is located on chromosome number 3, the name of the gene is the CNBP gene. Okay, CNBP gene which is located on chromosome number 3. If this gene is mutated, then that results in myotonic dystrophy type 2. Okay, so you have 2 types of myotonic dystrophies. In both of these myotonic dystrophies, what happens is that there is the muscular degeneration. Why there is muscular degeneration? Because there are 2 important defective proteins formed, which must be normal, but because of the mutations to these 2 genes, that protein which is formed out of these 2 genes, in this case we have the myotonic dystrophy protein kinase formed defective, and in this case we have the cellular nucleic acid binding protein formed defective. Both of the defective proteins lead to the degeneration of the muscles. I'll give you details about those things at the end of my lecture, because this is an autosomal dominant disorder and we need to focus upon the X-linked disorder, that is the Duchenne's muscular dystrophy and the Becker's muscular dystrophy. Currently, you need to remember one important thing. That means Duchenne and Becker, they are the X-linked recessive disorders. They belong to the muscular dystrophies, whereas myotonic dystrophy is not an X-linked disorder. It's an autosomal, that too inherited in a dominant fashion, autosomal dominant disorder. Itself, it's of two types, myotonic dystrophy type 1, myotonic dystrophy type 2. In both cases, there's an autosome involved, the mutation on the autosome. In one case, we have mutation on chromosome number 19. In another case, we have mutation on chromosome number 3. They are also very interesting that what type of mutations they involve. That's also very interesting. For the time being, just remember the name of the gene which is mutated. In this case, it's DMPK gene, and in this case, this is CNBP gene. I'll remove this particular dystrophy. Its effects, its symptoms, its appearance will be similar to what is happening over here, because these are also the muscular degeneration disorders. The difference is that the proteins involved and the chromosomes involved for mutation is different in myotonic dystrophy. That's very important. Now, my dear students, could you please tell me what is the job of a muscle? Could you tell me what is the job of the muscle? Muscles are responsible for helping us in contraction and relaxation. We are able to move from one place to another place once we perform the contraction and relaxation of our skeletal muscles. Our heart is capable of pumping blood all across the body because of performing contraction and relaxation of its 4 chambers. We're able to swallow things. We're able to, move the, swallowed material through our digestive system because of the contraction and relaxation of the muscles, smooth muscles which are present in our alimentary canal. So everything, be it the heartbeat, be it the peristalsis, be it the movement of the skeletal muscles, that is all possible because of the muscular tissue. Okay, so muscles perform the job of contraction and relaxation. Now, my dear students, if we look at, say for example, if we look at the skeletal muscles. If we look at skeletal muscles, a skeletal muscle is made up of these cells, what we call the skeletal muscle cells, or we call them the myocytes. They are, arranged in, parallel combinations, and each of these is called the myocyte, or we call it simply a muscle cell. If we try and look inside the muscle cell, suppose we take out one cell out of here and we just want to look what is inside the muscle cell, what is outside the muscle cell, there will be an arrangement like this, which is very beautiful in nature. So, muscle cell has got a specific nomenclature, you call it a myocyte. If you take into consideration the muscle cell membrane, you call it sarcolemma. This is the plasma membrane of muscle cell. If you take into consideration the endoplasmic reticulum of muscle cell, that you call sarcoplasmic reticulum. These are the basic things which you understand in class 11th. Similarly, if you take into consideration the cytoplasm of the muscle cell, you call that sarcoplasm. This is very important. So over here, if you try and notice, what is this blue line representing is the muscle cell membrane. I told you muscle cell membrane, you also call it plasma membrane, or you call it plasma lemma. Muscle cell membrane is also called sarcolemma. So if I look at this sarcolemma, the contents which are inside the muscle cell are on the bottom. The contents which are outside the muscle cell are on the top. Now what do you find inside the muscle cell? You will find important proteins. You'll find important proteins inside the muscle cell, what you call the contractile proteins. Could you name the contractile proteins, please? There are 2 important contractile proteins, what you call the actin and the myosin. So I have made over here a skeleton, a cytoskeleton of actin filaments. So this mesh is basically, this black colored mesh is basically the actin cytoskeleton. Okay, these fibrils basically contract and relax so that your muscle cell will contract and relax, and overall your muscular tissue will contract and relax. Okay, so what is inside the muscle cell is obviously the sarcoplasm, means the cytoplasm of the cell. And then you find this meshwork of actin cytoskeleton. Okay, what is outside the muscle cell is the extracellular matrix, which is also made up of fibers, and you have the fluid outside that's forming the ECF, what you call generally, and we can call it the extracellular matrix. It's basically, this matrix is rich in calcium ions. It's rich in calcium ions. Calcium is more in extracellular matrix. Now, my dear students, what else you get to notice over here is this complex. This basically is a proteinaceous complex which is present in the muscle cell membrane. This complex, like in your class 11th endocrinology, you must have studied about the receptors which are present in the cell membrane. Like you might have gone through the GPCR receptors or the protein tyrosine kinase receptors. They are the receptors which are present, which are embedded in the plasma membrane. So similarly, over here you have a glycoprotein complex. You have a proteinaceous body which is embedded in the sarcolemma. This complex is called the dystroglycan complex. Okay, if you notice, this blue color is the complex. Now notice this particular green color filament. This green filament is very, very important for you. This is the protein which is called dystrophin. This is a protein, very important for you people. What is this dystrophin protein doing? This dystrophin protein is having two ends, this end and this end. So this is the actin binding site of the dystrophin protein. If you notice, this is that part of the dystrophin protein which is bound to the actin filaments, and this over here is the dystroglycan binding site of the dystrophin protein. It has this region also. This is called the central rod. This is not a point of attraction for the time being. This part is the central rod part of the dystrophin protein. Now, if you notice the placement of the dystrophin protein, what is its job? You can think. What is the job of dystrophin protein? Suppose you think that there is no dystrophin protein, you will be able to notice that there will not be any sort of interaction between the actin filaments and the extracellular matrix. But once this dystrophin protein is over there, whose one end is attached to this complex and another end is attached to the actin, that is going to mediate, that is going to keep an eye on the movement of the the filaments as well as the membrane. Now see what is the job of this dystrophin protein. Once the cell membrane contracts, means once the nerve impulse reaches to the membrane and the membrane gets depolarized, and then what happens that the membrane has to contract or the muscle cell has to contract. Once the cell will contract, then there is a possibility that the membrane could rupture because it's a long cell. Once it will contract at one place, there is a probability that the next section will break off, but that will not happen if dystrophin is over here. Dystrophin is going to mediate the connection between actin and the complex, and this complex is in turn connected. It's associated, it's bound to the extracellular matrix proteins. So it makes sure that once the cell membrane contracts, at the point of then relaxation must be followed, and all these structures must come to their normal position without any damage to any of the constituents, specifically the membrane of the muscle cell. That should not be damaged. It's some kind of anchor which is going to maintain the integrity. It's going to maintain the, I will pen it down. So dystrophin protein is very important. This is what you got to notice from this diagram, that the dystrophin protein is important to maintain what? To maintain the integrity of the muscle cell. To avoid any damage to the muscle cell, to avoid any deformity in the muscle cell membrane, this is going to play a very crucial role, making it sure that once the muscle contracts and then it comes to its original position, there should not be any damage happening to the muscle cell membrane. Now, what happens? What is the possibility if you think like this happening, that there is the dystrophin protein which is not formed in a normal manner? Suppose the same dystrophin protein is mutated. Now, the question arises, if this dystrophin protein is mutated, means you don't have a normal dystrophin protein, you have a non-functional dystrophin protein. How can we show that non-functionality? Say for example, this was the normal dystrophin protein. Now you have a mutated dystrophin protein which is very short in length. That means the connection is gone. The connection is gone. Now you have a dystrophin protein which ends at this position. Think of the consequence. This is not a normal dystrophin protein. This is an abnormal dystrophin protein, you can say. AB stands for abnormal dystrophin protein. Think of the consequence. If now the membrane contracts, that means if it shifts, if it contracts somewhere, there's a possibility that this can break off. There's a possibility that this membrane can break off. Why has it broken? Why has it broken? It has broken because the dystrophin anchor is gone. The dystrophin anchor is not normal. Either it's totally absent or it is short, because of which if the membrane contracts, there is no anchor which is going to guard the reversion of the membrane at its normal position. So you will have these channels, you will have these gaps appearing on the membrane of the muscle cell. That's going to cause the problem. So see, look at now. Try and understand. Now you can think that the bond is no more, the integrity is no more. You can think of the consequence now. I told you that the calcium is more outside. Now there is a possibility for the calcium to enter inside. Once the calcium enters inside, it has to enter normally, but now more calcium will enter inside. More calcium influx will take place. Influx means the entry. More calcium entry will occur. This calcium has a job to do. There are specific proteins inside the sarcoplasm. Inside the cell membrane, you have specific proteins, what you call the proteases. Okay? Proteases. They are normally inactive. Okay? Now what will happen? This calcium will bind to the proteins. So this is what? This is the calcium which has bound to the protein. This protein is basically a protease. That means its job is to digest some proteins. Normally it has to digest those proteins. So I will show you over here that proteases normally have the job. These proteases normally they digest the non-functional proteins. They digest the non-functional proteins, means those proteins which are no more functional. They digest those. Now you have many such proteases active because of this calcium entry. Calcium has entered inside. Now more such proteases will be activated. When you will have more active proteases. So earlier it was inactive, but binding of calcium made it active. So now it is active. Active protease has the job to digest the non-functional proteins. But since you have more active proteases because of the entry of more calcium, now what will happen? Even these proteases will start digesting the normal proteins. Like your actin itself, actin as well as the myosin. That means these proteases will start digesting this structure. This will be gone. That means there is a damage happening to the contractile proteins. This is what you call the muscle cell degeneration. This is your muscle cell degenerating at this point of time because of the lack of this dystrophin, because of the lack of this, abnormal, because of the presence of this abnormal dystrophin protein, which has created a kind of imbalance in this mechanism, creating cracks in the sarcolemma, letting the calcium enter into the cell sarcoplasm, activating more and more these proteases. More proteases, once they are activated more in number, they will not make a distinction that we were supposed to digest the non-functional proteins. They will now start digesting the functional proteins, even the contractile proteins themselves, the actin and the myosin. That's why you call, at this point, what is happening is your muscle degeneration. We have written progressive muscular degeneration, because Almighty Allah has kept a mechanism of regeneration. Then again these proteins will regenerate, but at a certain particular point of time, when there will be so much the damage to the plasma membrane, a point will come when this entire myocyte will be gone. I'll tell you that also. This is one impact of this gap. Now there is another important thing which is going to happen. So one thing was calcium entering inside, activating the proteases, active proteases, damaging the important proteins of the cell itself. Okay, that was one thing which was happening. So what was entering was the calcium inside the sarcoplasm. So this is the consequence of the entering calcium. You have now understood. Now what else will happen over here is also very, very interesting. Inside the muscle cell, you have important molecules. I'll make these triangles. These are important molecules which are present always in the sarcoplasm. These are creatine kinase molecules. They are basically the energy packets, because muscle cells require a lot of energy for the purpose of contraction or relaxation. So creatine kinase molecules are always present in the muscle cell, and they donate the phosphate to convert ADP into ATP. These are normally present in muscle cell sarcoplasm, but via these gaps, these molecules go out. Okay. Once these molecules go out, so what are coming out now? These triangular molecules. These are what? These are creatine kinase molecules. Now, once their number increases outside, this becomes the diagnostic part. Creatine kinase level increases in blood. That is the part which is very important for diagnosing the Duchenne's muscular dystrophy. So creatine kinase levels in the ECF or in the blood will elevate once a person reaches to the prime of this Duchenne's muscular dystrophy. This is what is happening in Duchenne's muscular dystrophy. The same thing is happening in Becker's muscular dystrophy as well. The difference is only that Becker's muscular dystrophy is very slow process. Its progression is very slow. So it's slow progressive disorder, means the bad impact, the negative effects will not appear quickly. They will take some time, appreciable amount of time to appear, and it is not that severe. So it's severe, but it's less severe. It's not that severe. Which one is more severe is the Duchenne's muscular dystrophy. Otherwise, the same thing is going to happen in the Becker's muscular dystrophy as well. Now, what do we need to understand over here is that why is this dystrophin protein short? What has happened to the dystrophin protein? Why is it short? Why is it abnormal? Why is it not formed? Which is resulting in this bad thing, which is causing kind of a degeneration of the muscular tissue. That is to be understood. That means we have to go to the location of the gene which codes for dystrophin protein. We have to locate that gene, and we have to then say that either the gene gets mutated or not. Now, other thing which is very important to understand is that once this degeneration takes place, I'll show you the diagrams over here. So once the degeneration of the muscles take place, so definitely throughout the body the muscles will start degenerating, be it the shoulder muscles, be it the abdominal muscles, be it the leg muscles, be it the muscles of the arms. They will start degenerating with the passage of the time. Say for example, if you remember initially I told you that suppose these are the different muscle cells, okay? These are the different muscle cells. Now, suppose this muscle has started damaging over here. Suppose this muscle started damaging. It will repair for some period of time, but finally, this particular part will be occupied. This will be taken up by the scar tissue. That means the muscle is gone. It's filled by the scar tissue. So this is happening to your muscle. So your muscle is made up of myocytes. Now, one myocyte is totally gone. Not only one, similarly, other myocytes will also start degenerating with the passage of time. A scar tissue or either a fat will be deposited at this place. That's the point when your muscle will start losing its functionality. And with the passage of time, the entire muscle will be affected. Okay. Now, my dear students, this is the way in which you can think of the same thing happening to the shoulder muscles, the same thing happening to the skeletal muscles of the arms, of the legs, abdominal muscles, the pelvic girdle muscles. All of these muscles will be negatively affected. Consequently, what we will see in such individuals who are suffering from the Duchenne's or the Becker's muscular dystrophy, even for that matter the myotonic dystrophy, these individuals will not be able to stand up on their own. Till the age of 3 or 4 or 5 years, they'll be okay. But with the passage of time, because this thing is happening in their body, they'll be slowly losing their hold on muscles, and they will be in need of their arms to stand up. Once they need to stand up from the ground, they will use their arms to stand up. That's called the gowersign. Okay, look at the diagram. In this way they will stand up, and with the age of 6 to 7 to 8 to 9 years, they will not be able to walk on themselves. They will take help from the wheelchairs. That means they will start becoming physically disabled. Their legs will become, they will not be able to walk in a normal fashion. They will be a bit clumsy, and they have the chances of falling down because their muscles are not strong. Okay, and the other things which will happen is that their belly will protrude outwards because the abdominal muscles will be weak. Their shoulders will be drawn, they will be awkwardly placed, and they will be drawing their hands in the backward direction to walk, just to keep themselves in balanced position. Their lower leg muscles, they will become weak, and they will become fatty in nature. They will not be able to keep the spine in an erect position. That spine also becomes curved because of the damage happening to their muscles. So this type of, the image which is right now flashing on the board, this type of gait will be there. They will not be able to walk properly. So as far as the treatments are concerned, normally they are given the physical therapies, orthopedics give them the exercises to do. They are given the steroids to limit the degradation of the muscle cells. And they are more oftenly they are prone to the surgeries. There are different types of surgeries which are done to limit the damage to the muscle cells. And gene therapy is one of the treatment which could be thought about, although there is no such treatment available to the general population, because the cases of muscular dystrophy are very more. 1 in every 3,500 boys is affected. Boys. Okay, 1 in every 3,500 boys are affected by this particular disorder. Now let me give you an idea about what sort of mutation it involves. Okay, which gene is mutated. Now let us talk about the genetic mutation which is happening in muscular dystrophy. Specifically, we are talking about the Duchenne's muscular dystrophy. So you need to understand now. Duchenne's muscular dystrophy, and the same thing happening in case of the Becker's muscular dystrophy also. Becker's muscular dystrophy. Which is the gene which is mutated in both the cases is the gene for the dystrophin protein. So we'll write down the dystrophin gene is mutated. The gene which is coding for the dystrophin protein. This gene is mutated. Okay, dystrophin gene. Remember, there is one point which you can remember is that dystrophin gene is the largest gene in humans. This is the largest gene in humans. I have a question for you people. Tell me which is the smallest gene present in humans. The largest gene in humans is the dystrophin gene. It has around about 79 exons. Exons, the coding part of the gene. And the size is more than 2,200 kilobase pairs. Okay? The size is more than 2,200 kilobase pairs. This is the largest known gene which is present in the human cell. Okay. Now, my dear students, dystrophin gene codes for the dystrophin protein. What is the job of the dystrophin protein? That you people know, because that is going to anchor the dystroglycan complex with the actin cytoskeleton, and that's going to guard the inner contents with the extracellular matrix, which is very, very important. That is the job of the dystrophin protein. Okay. What is the location of the dystrophin gene is very important. Dystrophin gene is located on X chromosome. So if we look at the female has how many X chromosomes, please? Female has 2 X chromosomes. Okay. Suppose this is the location of the dystrophin gene. On the other chromosome, there will be other allele for this gene. So female has 2 alleles for this particular gene. One on one X chromosome, another on X chromosome. So these lines represent the 2 alleles of the dystrophin gene. Whereas if you look at the male has how many X chromosomes? Male has only 1 X chromosome, other one is the Y chromosome. Okay, so this is X chromosome, this is X chromosome, this is Y chromosome. So male has only one X chromosome. So if suppose this X is mutated, this dash represents the mutation in the dystrophin gene. That means the dystrophin gene is mutated. Okay, below line represents the mutated gene. And in this case, the other gene is normal. Let me show the other gene by X. So this is the normal case, okay. Here the female will not be affected because she has the other X to mask the function of this defective X. So this X will be producing the normal dystrophin protein. So this female will act as a carrier then, okay. She will not be affected, she will be only the carrier. But what if this male is affected? Suppose this dystrophin gene is mutated. So male dystrophin gene is mutated in a male. Male doesn't have the counterpart for this because this is Y chromosome. It doesn't have this allele for dystrophin gene. So male will be shown like this, okay. The other chromosome is the Y chromosome. So in this case, who will be affected now? Will this be a normal male or affected male? He will be an affected male who will be affected by the muscular dystrophy, to be specific, the Duchenne's muscular dystrophy or the Becker's muscular dystrophy. Affected by, let me write down the Duchenne's muscular dystrophy or the Becker's, because same thing happens in Becker's dystrophy as well, or the Becker's muscular dystrophy. They differ only in that this one is, Becker's one is less severe and it doesn't progress quickly. This one progresses quickly. Duchenne's muscular dystrophy progresses quickly. But if we talk about myotonic dystrophy, then X chromosome is not involved, okay. So that's why I told you earlier, if you remember, I told you 1 in every 3,500 boys globally is affected by Duchenne's muscular dystrophy. So this one is the prime disorder which we are focusing. 1 in every 3,500 boys. If we have 3,500 boys globally, one among them will be affected by the Duchenne's muscular dystrophy. Means his genotype will be X-Y. Now suppose I will give you a question. We'll take a female. So we'll be taking a female who is a normal female. So let me do a question. We have a normal female and we have an affected male. Let me see how is the inheritance pattern. It will be the same inheritance pattern which I have told you about hemophilia and color blindness. Normal female will be representing by XX, normal both, and affected male will be represented by X dash and he'll be having the Y, the normal, okay. So now the gametes over here, let me draw the Punnett square to show their children. Okay, so the Punnett square logic you people know. On this side we will pen down the sperms and on this side we will show the ova. So how many types of sperms the male will be able to produce? Male will be able to produce 2 types of sperms. One is the sperm carrying the defective X. Another will be the sperm carrying the normal Y. Ova will be of 2 types. We'll have 2 types of ova, the normal X and the normal X. Okay. So try and see what happens over here. The first individual will be shown like this. The second over here XY. This will be X normal and X dash. And this will be XY. Can you tell me what is the possibility over here? This will be a male or a female? This will be definitely a female, but again a carrier. Okay. There's an important thing to notice over here. This will also be a female, but she will again be a carrier. Okay. This over here will be the normal male. And this is also a normal male. What are you going to notice over here? That the affected male didn't forward this defect to the male children because father has to donate Y for giving birth to the male children. If father is affected, the trait will be transferred to the daughters only. So father gave this trait to the daughter and daughter has not become affected. She has become the carrier. Okay. son is totally normal. If this marriage happens, son born will be totally normal. Now, let us say, for example, marry this. Let us marry, say for example, the same daughter, this daughter. So we have the carrier daughter marrying a normal male. Try and understand. So we have a carrier daughter. How would we represent the carrier daughter? X dash with X. She is a carrier daughter. And the normal male we will represent by XY. This is a normal male. When this marriage happens, what is the condition of the children? Who will get the disease? Let me see. Okay. Let me again place the so over here I'll place the sperms and over here I'll place the ovas. So ovas will be of two types, X dash, and we'll have this X and X and Y. Now do a cross. This will be X, X dash. This will be X, X. This will be X dash Y, and this will be X, Y. Try and see what happens over here. Again, this will be a female affected or normal. She will be normal, but a carrier. It will be normal female. Am I correct? Okay. This will be an affected male. Affected male. And this will be a normal male. So, once the affected father, this was a couple, the father gave this trait to the daughter, she became a carrier. So father transferred it to the daughter and she became a carrier. Once this carrier daughter married a normal male, here we saw that the male, the son, became affected. So daughter transfers this trait to her son. Okay, son becomes affected. What do you call this transmission? You call this crisscross, or you call this zigzag, or you call it skip generation inheritance. So the father, if father is affected, he will transfer this trait to the daughter, and the same carrier daughter will transfer this trait to the son. So one generation gets skipped, this generation, this transference is called the skip generation inheritance. Now let me make you understand about what type of mutation is over there. Dystrophin gene is the largest gene, and if this produces the protein, if the same gene is mutated. Now what sort of mutation is that? Let me give you an idea, okay? Now, my dear students, let me make you understand about what type of mutation happens to this dystrophin gene, which is located on X chromosome. Dystrophin gene can either undergo the deletion or it can undergo the duplication. So basically, I've made you understand about these in my previous lectures. What is basically happening is a sort of frame shift mutation, due to which either the dystrophin protein is not formed, or it's malformed. Malformed, I mean to say, the length doesn't become the normal usual length. It becomes excessively short, as I demonstrated earlier. So overall, what is happening is that the dystrophin protein is, whatever is formed is basically non-functional dystrophin protein. Now say for example, I will give you the sequence just for the simplicity purpose, because I will not be able to show you the 2200 base pair long sequence. So what basically the gene codes, the gene has the information for the dystrophin protein, and the information exists in the form of triplet codons. So let me say for example, try and show the genetic code on the DNA. That means we are trying to show the sequence of the DNA. So this over here represents the gene. Okay, I need to rub this off. Okay, So suppose this is the gene, length of the gene. And so let me show you the codons in this case. One codon is made up of 3 nitrogenous bases. So at the end we have a special set of 3 nucleotides which is labeled as TGA. TGA is basically the stop codon. Okay, it's a stop codon. Okay, stop codon for the gene which is coding for the dystrophin protein. Now before that I will keep on adding the codons 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2. I'm making sets of 3, 3 because 3 nucleotides represent 1 codon. So I want to make it very specific. So this is suppose let me label it over here. So we have the suppose over here there is C, T, G, and then we have the A over here. Rest I'll make according to my choice. A, T, G, C, T, C, C, G, T, A, T, G, C, T, C, A, G, T, C, T. Okay. Say for example, this is the codon arrangement of the gene. So this is one codon, this is another 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 codon, and this is the stop codon as I've told you earlier. Now one codon codes for one amino acid. So say for example, this codes for one amino acid, this codes for another 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 amino acid, this codes for one amino acid, this codes for another amino acid, this codes for another amino acid. And these amino acids, let me tell you, suppose this codes for an amino acid, namely the, this codes for hat, this codes for cat, this codes for rat, this codes for mad, this codes for bat, this codes for fat, this codes for maybe the sat. Okay, suppose this is the arrangement of your dystrophin protein. So where is the information for this protein? Basically this is the protein. The hat, cat, rat, mad, bat, fat, sat is the protein, dystrophin protein, and it's for the formation of this protein the gene must be like this. Okay, now say for example due to some ill luck, or because I have already told you that mutations are spontaneous. Say for example something like this happened, that this particular C, suppose this cytosine undergoes the deletion. So there is just deletion of cytosine from DNA. And right now it's which gene? It's the dystrophin gene. This was basically which gene? From here to here. It was which gene? It was the dystrophin gene. Now in this dystrophin gene what happened? That this particular section got deleted. Could you please tell me what will happen to the gene? What will happen to the gene now? What will happen to the sequence of the gene on the DNA? Gene on DNA. What will happen? The codons will, ATG will be as such. ATG will be as such. CTC will be as such. CGT will be as such. Now could you please tell me what will happen because of this deletion? Won't be the entire reading frame after this mutation, won't that get disturbed? The reading frame will change. Now the polymerase is going to read, it was supposed to read C, T, G, but C is not over there. It will read T, G and A. Am I correct? So this will be paired with this. Then it will read T, G, C. Then it will read T, C, A. Then it will read it will read TCA, then it will read A, G, T. Then it will read C, T, T. Then it will read GA. At the end there will be G and A. I have written over there. Now see, this is the same thing, this is the same thing, this is the same thing, if I'm correct, but this is now totally changed. TGA. Could you tell me what TGA is? TGA is the stop codon. Could you tell me what this has the information for? This will code for the, this will code for hat, this will code for cat, and then the chain, the protein. Which protein? This was dystrophin protein chain will end. Is it ending at the normal position? No, it will end. It will not be formed completely. We will call it a premature protein. It is a prematurely formed protein. It will not be of the usual length. It will not be of the normal length. It was supposed to be of something like this nature, but now it will be excessively small in size. That's what I told you earlier. The dystrophin, mutated dystrophin protein is short and it will not be able to bind to the dystroglycan complex. Consequently, the short dystrophin protein will not be able to fulfill its job. What is this happening? Deletion. Once deletion is happening, which type of mutation is happening? Is the frame shift mutation. Which mutation is this? Frame shift mutation, because the reading frame of the gene is changed. Once the reading frame is changed, you get some stop codon appearing. You find out some stop codon appearing. This was not the initial case. Earlier it was CTG codon, but now what appeared was TGA codon, because this got paired with this got paired with this, and they formed triplet like this. The entire frame, the reading frame, has now changed. Consequently, the dystrophin protein, which was to be formed, the dystrophin protein got mutated. That will result in the Duchenne's muscular dystrophy and the Becker's muscular dystrophy, which I made you understand earlier, that once the dystrophin protein will not be normal, the calcium will travel in through the leaks in the plasma membrane, and that will activate the cellular proteases, killing the normal, damaging the normal actin and myosin filaments. Muscle cells will start deteriorating at a very fast pace. So just by reaching the age of 15 to 20 or maximum 30 years, the individual dies, because they cannot live the normal span of the life. Their muscle cells are degraded at a very faster pace. Okay, this was all about the Duchenne's muscular dystrophy and Becker's muscular dystrophy. As far as the myotonic dystrophy is concerned, let me give you a slight idea about that as well. Although I have told you about that thing, that in case of the myotonic dystrophy, X chromosome is not involved. Number one thing, the mutation is on the autosomes. If there is chromosome number 19 involved, then you have a gene, what you called the what is that gene called? Is the DMPK gene. DMPK gene basically has a repetitive sequence in it. That repetitive sequence is a sequence of CTG. That this is a repetitive sequence which is present in this gene. If this CTG enhances in number, if it's present in more number, that means the CTG trinucleotide repeat. If its number increases, then DMPK gene gets mutated. In that case, there's a protein which is called the
dystrophy. Rather, we call it myotonic dystrophy.
Protein kinase. That is not formed in a correct fashion. When this protein is not formed, when this protein kinase is not formed, that results in the muscle degeneration. Similarly, we have another important thing, because I have told you initially that myotonic dystrophy is of two types. This one is which myotonic dystrophy? This one is myotonic dystrophy type 1. We call that DM type 1. Okay. Similarly, we have myotonic dystrophy type 2, in which the mutation happens to the chromosome 3. On chromosome number 3, you have a gene which is called CNBP gene. CNBP gene also has a repetitive sequence in it. It has this has CTG sequence, it has CCTG sequence. It's a tetranucleotide repetitive sequence. If this repetitive sequence increases in number, then CNBP gene is mutated. And once CNBP gene is mutated, it is responsible for producing a protein which is called cellular nucleic acid binding protein. Then this protein is not formed. Okay? because of the mutation. Repeat is present normally, but if the number of the repeat increases because of the duplication mutation, then this protein is not formed. And once this protein is not formed, it again results in the muscular degeneration. This type of muscular degeneration or myotonic dystrophy is called DM type 2. Okay, so you have DM1 type 1, DM type 2. In both cases, you saw autosomes involved, chromosome number 13 or chromosome number 3. Since autosomes are involved, we do not consider myotonic dystrophy as an X-linked recessive disorder. It's an autosomal dominant disorder. The effects will be same as the Duchenne's or the Becker's muscular dystrophy. This was all I had for you people in this video lecture. If you find it interesting, please give it a like, a share, and a comment upon my video. And stay tuned, inshallah. I'll be coming back with another important, genetic disorder. Inshallah, hopefully I'll be taking up the lecture for sickle cell anemia. That's going to be very interesting. That will be coming up soon. Take care of yourselves. Please remember me in your prayers. Fi amanillah.